Thank you for that wonderful introduction. Yeah, sometimes when people introduce me, I can kind of sit and look, maybe they're actually talking about someone. But yeah, thank you very much. So today I would share a part of my research, and this was mainly the research I carried out during my PhD. So I studied mainly the common bulbul and tried to use it to understand how birds cope with changes in rainfall pattern and seasonality. And before I go into the talk proper, I would want to acknowledge the following people who were instrumental to the entire package, including supervisors and people who assisted me in data collection, and master's students who joined the project at some point in time. So this is something I wrote at the end of my PhD, and I thought it's important and to share because so I thought that when you study a species that is common, you are able to engage with the ecology of the species properly. You can focus on a biological question, and rather than spend a lot of time looking for your study species, because when I started working on the common bulbul, so many people were surprised that why the common bulbul, what is interesting about the common bulbul. But what's interesting is that you find it everywhere. They breed throughout the annual cycle, and you can get a good sample size to test really interesting biological question. So when you think about study, sometimes we are carried away about very interesting species, and we can spend the whole we can spend the entire study period trying to get a sighting of that species. So this is something to think about. So let's consider a few background concepts about the entire study before I then go into the four different things that I will talk about for this presentation. Our tropical environment really seasonal, because there's that general notion when people talk about the tropics, they think about this environment where there is food year round. There is where there's 12 hours of light and 12 hours of no light, and conditions are just almost exactly the same throughout the year. So you find people making such sweeping statements saying, oh, the temperatures are seasonal, the tropics are stable. But we know clearly that in some tropical systems, like in the savanna, we have a clear dry season and a clear wet season. And in central Nigeria specifically, and some other parts of Africa, we are likely to have a period of 12 calendar we have like 6 months without rainfall completely. And then once the rain stops, the environment gets drier and drier and drier, and the peak of the drought is just before the next rain starts. So that's as best as seasonality can get. So it's just a different factor driving environmental seasonality. And this is similar similar to what you see in the temperate system, where you get the very cold part of where there is snow and all that, and then in the summer where the environment becomes then conducive again, and people can go around without any jackets. Okay, looks like the slides are a bit stuck. Okay, fine. So, and if you look at seasonality too in a quantitative way, this is 2 years of rainfall data from Jos, central Nigeria. You see that you have about 6 months here of rain, and then about 6 months without rain, and another 6 months of rain, and then 6 more months without rain. And also the mean diurnal temperature pattern also shows this very clear seasonality. So this is as seasonal as it can be anywhere in the world. And then if you look at seasonality too from the perspective of the common bulbs, we see clearly that for breeding is likely to take place at any part of the year. This is 2 years of breeding data based on brood patch occurrences. And you can see that you can practically see birds breeding at different parts of the year. Whereas an annual cycle stage like molting, which is the change in the feathers, it's very clearly seasonal. In every year, there's a part of the year where birds change their feathers, and that is specific. So while some annual cycle stages might not be as seasonal, some annual cycle stages are clearly seasonal. So even from the perspective of the birds, you see seasonality in specific annual cycle stages. So for those who are not very familiar with changing of feathers, which is called, you see clearly that these are new feathers and these are old ones, and there's a sequence of change from this to this to this and then to this one, and this process is called molting. So let's look at the annual cycle now properly. What do we stand to gain by studying birds that breed throughout the year? If breeding activities occur at the different parts of the year, you are able to find birds in wet conditions, in dry conditions, and so you can actually test how these environmental conditions affect breeding. While things like other annual cycle stages like molting takes place at one part of the annual cycle. And then you find resources like food, fruits, and seeds, and insects. And also you find pathogen pressure also varying depending on the environmental condition. And this is different from, very different from a temperate system where breeding takes place at only one part of the annual cycle, in spring going into summer. And that is the same time where you have a lot of food in the environment. And so at the same time, you have high productivity, so you can have high pathogen pressure. And so if you find, so it's not easy to say that it's either the resources, the temperature, or the lack of snow that is responsible for whatever you see, because it's only seen at one part of the year. But under this system where breeding is taking place at different parts of the year, you can decouple these different environmental conditions and test which ones influence the trait of interest, and in this case, breeding. So let's look at, let's now consider immune function. Generally, we understand that the immune system is built to protect animals from getting sick. But at the same time, but at the same time, running the immune system actually incurs cost. So there are two hypotheses in ecological immunology when people study the immune system or study immune function. One is the antigen response hypothesis. And there, we predict that immune function should vary with infection risk. So if infection gets higher in the environment, then the immune function should also be high. And then you have the resource constraint hypothesis. That there we predict that the immune function should vary according to resource levels in the environment. So if there's a lot of food and there's enough to invest in both, and there's enough to invest in immune function, then immune function should be high. But where you don't have enough resources, then animals are likely to down-regulate immune function. But we'll see whether this actually holds every time. But when you then look closely at immune function, you realize that it's actually much more complicated, because because every animal needs to go through very tough seasons and deal with several things. And it's not just one thing that constitutes a difficult time, both infection risk, changes in temperature, changes in food availability, and so on. And all this form a package that you see as environmental change. But also, there's also variation at the population level and changes that also take place at the individual level. And immune function sits at the center of all this. This illustration here is just to show how complex studying variation in immune function can be. And we'll see how this relates to coping with wet and dry seasons in a species that breeds throughout the year. I carried out my study in Jos, central Nigeria, so around the AP Leventis Ornithological Research Institute. And one exciting thing about this system, like I said earlier, is that you have a wet season and a dry season. You can see the clear contrast between environmental condition when it's raining and then when it's dry, the environment is almost totally dry. But then one thing you would see is that even in the core of the dry season, you still see that some plants are able to maintain some greenness, which means that it's not all dry completely, or even when it's completely wet. So you then local conditions and specific changes become very important to in explaining how birds deal with environmental change. And I'd like us to keep that in mind. So my study was generally broken down into three main parts. First, I considered how birds fit in non-seasonal breeding with moult, because we expect that annual cycle stages should be separated in time, because you don't want to be investing in two costly annual cycle stages at the same time. So if a bird is likely to breed throughout the to breed both in the wet season and in the dry season, then when does it fit in when does it fit in time for molting? Then I also considered what explains seasonal variation in immune function. is it changes in environmental conditions like dryness, whether it's raining or not, whether there's rain or there's no rain, or actually whether it is better explained by whether a bird is breeding or not breeding. That is variation in the timing of annual cycle stages. Then after looking at immune function variation in time, we then checked in space whether the patterns we see in time are similar to the changes we observe in space, because aridity actually varies both in time and in space. And Nigeria has an important, very clear rainfall gradient from the north to the south. Then we picked up one aspect of, we picked up one factor, which is diet, that can vary in the environment, and we manipulated it in captivity to test how this change can affect immune function. So quickly, we went out and we tried to catch a common bulbul every day for 2 years. So for every bird we caught, we took body mass, we collected some blood samples, we checked whether the bird was molting, and we checked whether the bird was breeding. And we had this data for two years. And when we plotted the data, and we also aged the birds too, to know whether they were young birds and old birds. And when we plotted the data, we see clearly that birds can actually breed at any point in the year, even though there's some kind of a small peak here and here, which is actually a bit circular. But then we saw that birds actually molted at the same time in the year every year, suggesting that despite year-round breeding, birds still molted at the same time. So the year-round breeding doesn't prevent the birds from maintaining molt seasonality, and molt was restricted in the wet season when conditions are better in the environment. And this suggested that for this species that is able to breed at any time in the annual cycle, molt might be a much more important annual cycle stage, because molt is directly related to adult survival. And if you think of it this way, most breeding attempts in the tropics get predated, especially for open cup nesters like the common bulbul. So if your nest is likely to get predated and you continue to try, it might be more beneficial for you to invest in self-maintenance. And by doing so, you're able to survive and breed again in when the opportunity presents itself for breeding, rather than try to fit in breeding to the best environmental condition, when no matter how much you try, you cannot guarantee whether your nest will not get predated. So for a species like this, self-maintenance may be an annual priority compared to investing in reproduction. But just to check that we didn't have distinct breeding populations, so that say we had like dry season breeding birds and wet season breeding birds, and that we're doing that consistently, because we caught individuals. 25% of the birds we caught throughout the entire study period were birds that we were catching for the second or the third or fourth time. So we're able to check whether it's the same individuals that were breeding across the year. And you can see clearly that this is individual flexibility. It was not as if we had a population breeding in one part of the year and another population at a different part. So individuals can breed at any part of the year. And we were able to know this because we coloring the birds and we can see the same individual breed again and again within the same year. But how do birds cope with breeding in the wet season and the dry season? We thought that there must be some kind of a mechanism that allows them to cope with the harsher breeding condition in the dry season. And what we found out was that the birds that bred in the dry season actually put on a higher body reserve. So they were heavier than the birds that were breeding in the wet season, because the dry season is much more unpredictable. When you actually carry body reserves, then you can spend more time incubating, and then when you don't find food in time when foraging, you would not starve. So this gives us some idea about how birds try to kind of cope with breeding both in the wet season and in the dry season. But more importantly, it tells us that the birds need to use the best time in the environment to prepare to actually prepare and be in good conditions, be able to deal with conditions in both the wet and the dry season. So let's wrap up this part. And what we can conclude from here is that individuals take breeding opportunities like throughout the year, but then they're able to kind of adjust their body condition in such a way that it allows them to deal with the environmental conditions that are not the same. But then despite breeding being non-seasonal, change of body feathers actually take place at the same time every year. And so that it suggests to us that there's no causal link between breeding and molting. These events are likely independent in the annual cycle. So let's quickly go into immune function. So what explains variation in immune function? Remember, I talked about the antigen response hypothesis and the resource constraint hypothesis. Under these two hypotheses, you have different predictions. If we assume that the wet season, which is highly productive, should have a higher pathogen risk, then we might expect birds to invest a lot in immune function. And so immune function should be high in the wet season and low in the dry season, regardless of whether you are breeding or not. However, if resource constraint becomes important, then you have three different possible scenarios, and this can lead to different predictions. I won't go into the details of this, but what this does is that the difference you see between breeding and non-breeding birds would depend on whether resource constraint is an issue in the wet season or the dry season. So what we did for this part of the study is that for the blood samples we collected, we measured lysis, and lysis is complement activity, and we measured haptoglobin concentration, which is a measure of inflammatory response. Here, what we do is we take the plasma, 25 microliter, and we incubate in serial dilution with 1% rabbit red blood cell and incubate and test how much concentration of the plasma destroys the rabbit red blood cell. So you can look at it as a very simple way of testing how the bird's plasma deals with foreign cells. So the higher I mean, the lower the concentration that destroys the rabbit red blood cell, the stronger the immune function of that individual, the stronger the immune function of that individual bird. Also, for haptoglobin concentration, which is a spectrophotometric assay, we take a quantity of plasma and run a test. And the higher concentration of haptoglobin you find in the plasma, the higher the inflammatory response. So you would expect that if an individual bird is responding to infection, the haptoglobin concentration will be high. Whereas for hemolysis titer, if you see a higher lysis titer, you might interpret it as the bird being capable of destroying foreign cells. So just to give you an example of the score, this you would score as a 4, and this you score as a 3.5 because there's a partial destruction of the cell, and this you score as a 1.5. So we did this for about 850 samples that we collected in 2 calendar years, and we have these results. So if you see here, you notice that what we find is that immune function was actually predicted by season rather than the annual cycle stage in which the bird was in at the time we trapped them and collected blood from them. So both lysis and haptoglobin concentration were higher in the dry season compared to the wet season, regardless of whether birds were breeding, molting, or not doing any of the two. And when you look at this pattern also within individual birds, you see that this pattern that you see at the population level also takes place within individual, which means it's an individual response rather than difference among individuals, because these were birds that were caught in the dry season and in the wet season. And you can see that for all the birds you see here, the haptoglobin concentration was higher in the dry season compared to the wet season, and so is lysis titer. I know you are concerned about the outlier here, but this is actually to make the point about this sample was collected in April, and in central Nigeria, the wet season starts around April and ends in October. So it now calls for that problem where do you draw the line between the wet season and the dry season? It's always variable. Sometimes the rain starts early, sometimes it starts late. So this bird that was caught in April was probably still in dry season condition, and that's why you have this high lysis titer. So you would notice that for immune function to be high in the dry season compared to the wet season is contrary to what we predicted, because we expected immune function to be higher in the wet season when the environment is much more productive and the pathogen pressure is expected to be high. So we expected more ticks and more mosquitoes, more malaria, more bacteria to be more abundant in the wet season, and this should make birds sick. But we don't see that, at least from immune function, we don't see that birds were likely to be dealing with higher infection risk in the wet season. So what we thought might be the alternative hypothesis is that perhaps what is important is that in the dry season, different species cluster around fewer resources. And in the dry season, you don't have a lot of flowing streams because there's no rain. And so there might be higher risk of depositing pathogens in the water bodies that don't flow. And different species come together, and that might lead to transmission. And so it might not be the abundance of pathogen in the environment, but the risk of transmission between individuals. And this is also a period in the year where resources are slim. So birds might be in poorer condition, and so might need a stronger immune function to be able to go through this tough season and survive for the next breeding season. So it means that predictions of variation in immune function cannot just be simple. We must take into consideration the environmental context and the life history context of the species. But then when we looked at the species that were breeding in the wet season versus those that were not breeding, all within the wet season. So looking at birds under the same environmental condition, we actually saw that the birds that were breeding for lysis titer had a higher lysis compared to the birds that were not breeding. But for the other immune assay, which is haptoglobin concentration, the birds that were not breeding actually had a higher haptoglobin concentration compared to those that were breeding. This was a bit confusing, but then if you think about it this way, perhaps you need to be in good immune condition to actually breed. So then the birds that are not in very good condition are or don't have the capacity to deal with infection actually skip breeding. So self-maintenance might be very important for this species, because generally we only find very few species breeding at any point in the year. So your physical condition and your physiological condition might actually be what triggers you to breed. These were part of the insights that took me to ask the question, why do birds breed when they do? And when you look at the haptoglobin concentration, it suggests that the birds that are responding high to inflammation, so birds that might be dealing with infection, are the ones that are not breeding. So if you put these two together, it means that birds breed when they are in the very good condition, and birds that are not in very stable condition just skip breeding until when they are in better condition. Well, that's a speculation from what we see here. So we can say that environmental seasonality actually predicts immune function better than annual cycle stages, at least from the common bulbul. Weather conditions do not necessarily imply high infection risk, and the occurrence of so when birds actually change their feathers, it doesn't make them to down-regulate immune function. So we cannot argue that that immune function is down-regulated because of the cost of changing feathers. And when this is done in very good condition, birds can maintain maintain their immune function at a good level, as well as change their feathers to be prepared for harsh conditions. So then, after seeing all this difference in time, we decided to check over an aridity gradient in Nigeria. If you've been through Nigeria, you will realize that there's a fine gradient of environmental condition from the northern part of Nigeria, which is just around the edge of the Sahel, and the southern part of Nigeria, which is on the coast of the Atlantic, Ocean. So this part is quite wet, and this is very dry. We went from this location down to this location in from the end of January to the first week of April. And the reason why we did that was because the rain starts early in the south and later And so by starting here, we are able to go all through to the from the south to the north before the rain starts. So all the locations were sampled before the wet season. Our interest was to see whether, since we found a clear difference between the wet season and the dry season, wanted to see whether immune function was also higher in drier compared to wet areas, or in wetter compared to dry areas when we look at variation in space. So we did the same thing we did for the first study: collect blood samples, measure lysis and haptoglobin from the 15 locations. Interestingly, there was no trend from north to south or from south to north. You see that from the very cool and wet part of the country to the very hot and dry part of the country, both for haptoglobin and lysis titer, we did not see pattern. But what we see is variation among individuals within each of the locations where we sampled birds. So what drives variation is in immune function might be very local. Differences among individuals where they are, or kind of short-term changes in environmental condition within each location. So birds across the Arctic region are able to maintain their immune function differently based on their circumstances. But then, because we sampled the center of the gradient twice, we're able to test whether there was already a change in time. And for haemolysis titer, we already see clearly that between the last week of January when we started and the first week of April when we completed the study, there was already a difference in lysis titer between the two periods in the year, reinforcing the fact that the variation in immune function, which we saw in time in the first study, was not just a fluke. So we can conclude from this part of the study that variation in innate immune function does not follow spatial aridity gradient. The pattern we see in time is not consistent with changes in aridity in space. But then we see clearly that there's huge variation within population. So individuals do different things, even under the same environmental conditions. Okay, so now the last part of the study is about diet. Excuse me. So now what we did here was we isolated one component of the environment, which is food, because food resources vary across the different parts of the annual cycle. Birds get a lot of fruits in the wet season, maybe not as much in the dry season, and different amounts of insects throughout. And depending on whether the environment is degraded or not degraded, they also have different resources available to them. So what we did was we took birds into captivity, 40 of them, 20 in one group, under 20 in the second group. We fed one group with fruits and the other group with insects or invertebrates generally. And this included meal, we got like grass hoppers, termites, and all that, and just fed them with it. While one group had exclusively fruits. And these fruits included fruits we bought from the market and fruits we actually collected from the reserve that were in season. And after 12 weeks of feeding birds, we took half of each group and switched to the alternative group and continued for another 12 weeks. Throughout these 24 weeks that this experiment took place, every 2 weeks, we caught all the birds there, collected blood sample, took their body mass, and then checked whether they were molting or not. And we returned them back into captivity. So we did the same measurements of immune function, we checked whether birds were molting or not molting. So we had physical condition, which was like their mass, and whether they had said like changing their fed the others, and then we combined the immune assays that we measured into a principal component. And I'll quickly explain. You would realize for the previous 2 parts of the study involving immune function, I used haptoglobin concentration and haemolysis titer separately. But for this study, we decided to combine them into one component and call it immune function. Here, you will see that as the PC1 increases, it means an increase in haemolysis titer. However, as the PC actually increases, it means a decrease in haptoglobin concentration. So a high PC PCA here would actually mean high haptoglobin high hemolysis titer and low haptoglobin concentration, while a low PPC means high haptoglobin concentration and low hemolysis titer. So what did we find when we looked at the results? You see that before the experiment started at week 0, there was not a significant difference between birds that would be fed on fruits and birds that would be fed on invertebrates. However, 12 weeks into the experiment, you see that the birds that were fed on fruits had a higher immune function compared to birds that were fed on invertebrates. These groups were then split after 12 weeks to continue for the next 12 weeks. And you can then see that the birds that were moved to an invertebrate diet from the general fruit diet actually reduced their immune function. And those that were moved from the insect diet to the fruit diet actually increased their immune function, to show that what is driving this change that we see is diet. A fruit diet is clearly better for the immune function compared to an invertebrate diet. And if you look at the body mass too, you see exactly the same thing. No difference when the experiment started, a clear difference between fruit-eating birds and invertebrate-eating birds, and a switch when you divide them. Then the last thing was the onset of moult. As at the time where we ended the experiment, which is the second week of May, for the common bulbul at the population level, moult starts on average at the 5th of May each year, plus or minus 4 days. So at the time where this experiment ended was just about when the birds were starting to change their feathers. And you can see clearly that it was only the birds that were on a fruit diet at that time that had commenced molting. The birds who were on the invertebrate diet had not started molting completely. And just to add to that, it was until we introduced the birds on the invertebrate diet to a fruit diet that moult started. That's at the end of the experiment. So the guys here. So but then we tested one last thing. You remember I said in the beginning that when resources are slim, birds are likely to trade off immune function for other annual cycle stages like breeding or other aspects of self-maintenance. But we see clearly from the results that the birds that were in quality conditions, as birds that were fed on fruits, were able to maintain both high body mass and also a high immune function. So it means that actually the trade-off between immune function and other annual cycle stages or other processes depend on how much resources are available, which actually makes the resource availability hypothesis actually valid. But they are not just two things that take place at any point in time. There are several other things that are competing for resources at any point in time. But then if you look at the birds that were fed on invertebrates that were in poorer condition, and so anytime they increase the immune function, you're likely to see a decrease in body mass. And this is likely because the muscle tissues are broken down to produce acute phase proteins when resources are not sufficient. So under low resource conditions, when you have a resource constraint, it is likely that you see a clear trade-off between immune function and other processes. Within the little part analysis, we try to find out whether the diet manipulation affects immune function directly or indirectly. And we can see from here that the diet affects haptoglobin concentration specifically, and increased haptoglobin production has a negative effect on body mass. And that actually reemphasizes the fact that body tissues are broken down to produce acute phase proteins for immune function. So we see that diet actually affects both body condition and several other both immune function, body mass, and the onset of molting directly. And this happens rapidly. So if the environment is altered in such a way that diet becomes affected, birds are likely to respond immediately. But here we kind of get the idea that because common bulbul are able to find fruiting plants and resources generally throughout the annual cycle, and their ability to use different resources at different parts of the year, this might actually kind of buffer these harsh conditions that they may face throughout the year. So the availability of resources might be an important factor that allows them to maintain that year-round breeding strategy. So let's go back to this complicated diagram just once more. We see that the environment actually plays a huge role in predicting immune function, variation in immune function and other annual cycle stages. We see that individuals also show a huge difference. So the local condition of an individual or the circumstance of an individual might really be important in what you see. But at the population level, we don't see this huge differences. So it is not likely that the annual cycle stage, say we have a breeding season at the population level, and then a molting period, that we see this clear difference in immune function. Individuals actually responded according to their circumstances, but the environment actually played an important role. Some kind of what we might call even an overriding influence on the annual cycle stages. So what do we, so putting everything together, like we looked at breeding, immune function, body mass variation, moult, and all the seasonal variability. What do we take from there? We see that immune function might actually be a very important factor that allows birds to cope with the wetness and the dryness that takes place seasonally. And by birds being able to maintain their immune function in such a way that helps them throughout the year, they're able to cope with the seasonal rainfall and still able to maintain breeding. And it looks like the timing of breeding has little fitness consequence on this species because the birds can actually just breed at any time in the year when the opportunity is there and their condition is suitable for breeding. But then it seems to be that there's a very important time in the year where birds have to actually change their feathers so that they're in good condition for the harsh season. So to interpret whether immune function is costly or not, we can only interpret that within an environmental context or within a life history circumstance. So we cannot just say investment in immune function should lead to a decrement in body mass or should make birds stop breeding. The individual circumstance is very important. So like the trade-off between immune function and other annual cycle stages is not just black and white. So any assumption of disease risk needs to be tested empirically. Remember in the beginning, I predicted, like I explained to you that our prediction when we started this study was that immune function will be high in the wet season because we expected a lot of more pathogens, vectors, and nonsense and all that. But clearly we see that it is likely that the interactions in the dry season and the other and the local conditions that birds might experience in that season might actually be responsible for lowering or raising infection risk rather than just high productivity. And life history trade-offs do not always appear as negative correlations between factors. Usually we expect one event to decrease if the other is rising, because we also always say that one resource cannot be invested in two things at the same time. But one thing we need to understand is that it's not just those two things that we measure that are happening. Several other things are happening at the same time. And if birds are in quality condition, we might not see any decrement in the two things that we are measuring. And finally, from our diet experiments, we see clearly that it doesn't look like proteins are the main limiting factor for the immune system, because we see that the birds that we fed on invertebrates were the ones that did even that did badly compared to those we fed with fruits. So it is likely that good nutrition actually prevents you from even being susceptible to infection. And this you see in wild-caught adult birds that had already been adapted to wild conditions. They were just brought into captivity and manipulated. So you can see that diet might play a very important part in helping birds to maintain proper condition and year-round productivity and breeding in the wild condition. So I would like to also thank these organizations where I got most of the funding for during my study, and to everyone who has listened patiently. I'm happy to take any questions that you might have. Thank you very much.